Welcome students to chapter 3, which is about cells. So we're essentially kind of moving up the biological hierarchy. Chapter 2 was about atoms and molecules, which is kind of the lowest level on the biological hierarchy. And then when we put atoms and molecules together, we get cells and organelles. So this particular chapter is broken into 3 parts. We will actually spend most of our time in part 1 about the plasma membrane and part 3 about the nucleus. Part 2 is the cytoplasm, and it's mostly kind of the metabolic machinery of the cells, and there's just a fantastic table in your book that kind of covers all of it. But where most of the kind of action happens in terms of how cells interact with each other, and then how they repair themselves, and how they contain our DNA, and how our DNA is kind of the basis for everything, is then in part 3. So most of these lectures are going to focus in part 1 on the plasma membrane and part 3 on the nucleus, but we will briefly talk about part 2 as well.
You and your future patients are each made up of trillions of cells, so it's crucial to understand what cells are and how they work. In this chapter, you will learn about the general structure and function of cells. There are over 250 different types of cells in the human body, but all of these cells include 3 main parts: the plasma membrane, the cytoplasm, and the nucleus. The plasma membrane is the outer boundary of the cell, which contains the cytoplasm, and the nucleus, the cell's control center. The plasma membrane of a healthy cell is a selectively permeable barrier. This means that the plasma membrane allows beneficial substances like nutrients to enter and remain in the cell, while keeping undesirable substances out of the cell. It also keeps valuable proteins within the cell and allows waste to exit. When the plasma membrane is severely damaged, like in severe burns, the cell becomes highly permeable and substances flow freely in and out of the cell. For example, this is why severe burns weep, as proteins, ions, and fluids leave the damaged cells. Burn dressings are key for protecting these damaged cells from the environment. A thorough understanding of cellular structure and function will enable you to properly treat all of your patients, including those with cellular diseases and cell damage.
So as the video mentioned, we have over 250 million types of cells in our body. You can see from this diagram here that we've got a huge variety in terms of our cells. We can have essentially small little sacs of an oxygen-carrying pigment called hemoglobin in the form of our red blood cells, our erythrocytes. We can have these long, cylindrical, kind of contractile cells in our muscles. We can have these globular fat cells and these kind of globular kind of Pac-Man cells called macrophages that eat and that essentially eat disease and bacteria. We can have these neurons and neural cells that have these long kind of arms and processes that hang off of them for kind of transmitting information throughout the body and controlling the body. We have epithelial cells and connective tissue cells that make up our bones and our cartilages. So we have a huge variety in terms of our cellular structure and function. But the reason we start with cells before kind of moving on to all of the organs is without our cells, we're kind of just nothing, right? All of the amazing things that our body can do is all dependent on how our cells function. Now, all of our body cells start off very similarly, but then through the process of transcription and translation, which we'll talk about in a little bit later lecture, we get this huge, this just amazing variety. Even though all of our cells share the same DNA, different parts of our DNA get activated, and that's what then the process of differentiation results in this kind of huge variety. Now, one thing that all of our cells will have in common outside of the shared DNA is that all of those kind of necessary life functions that we went over in chapter 1, the idea of responsiveness and metabolism and digestion and growth and repair and all that, like, a cell is the smallest kind of living level where we actually carry out those processes. Cells have a metabolism. They digest things, they excrete wastes, they respond to other cells, and so kind of the smallest unit of life characterized by those processes that we talked about in chapter 1 is here at the cellular level. And so whenever you hear somebody talking about cell theory, that's kind of what we're talking about. It's this idea that even though we have this great variety of cells, they all share the same DNA per person, but through differentiation, we get a huge variety in terms of structure and function, but that the smallest unit of life, the smallest unit that can carry out processes that characterize life, is here at the cellular level. So if you've ever had a biology class, probably ever, then you have probably seen kind of the generalized kind of human-animal cell diagram before, right? So our cells have 3 basic parts. The membrane, which is that outer boundary made up of phospholipids. The cytoplasm, which is the stuff inside the cell, the fluid plus all of the organelles, the machinery of the cell, and then the nucleus. It's kind of like the control center, the brain for each individual cell. So just like our brain is kind of the control center for our whole body, the nucleus is the control center for each individual cell in our body. So this is the kind of generic animal cell diagram. You can see here's the plasma membrane of phospholipids. You can see we have mitochondria. That's a really important organelle in the body, and actually was once thought to be a free-living organism that got subsumed and eaten by a larger cell. But so it actually is quite unique in that it has its own DNA, which the other organelles in the body don't. Mitochondria are our cells' kind of powerhouses. They're the generators. They make ATP so we can power our body. You can see the nucleus here with all of the kind of what we call fluffy chromatin, which contains our DNA. We also have structures that help us build proteins called ribosomes. Some of those ribosomes are attached to the endoplasmic reticulum, and some of them are kind of free-floating in the cytoplasm. We also have a smooth endoplasmic reticulum that plays more of a role in synthesizing lipids and other kind of fatty types of molecules. The Golgi apparatus here is a kind of like a warehouse. So stuff that is made by the rough ER kind of gets then sent to the Golgi apparatus so that the Golgi can kind of repackage it and make sure that any modifications that need to be made can be made, and then it kind of sends it out through what we call a vesicle, which is essentially just a sac of phospholipid membrane containing either some sort of secretion, some sort of proteins that are made, or maybe something that's coming into the cell. So you'll also see lots of cytoskeletal elements, some of which, like these centrosomes, that play a role in mitosis and growth processes, others that give kind of shape and structure and kind of guide wires for the cell. We also have 2 kind of recycling types of organelles. That's these lysosomes here and these peroxisomes. So lysosomes contain acidic enzymes. Peroxisomes contain, what do you think? What does that word make you think of? Peroxisomes, right? Peroxide, perhaps? They contain actually peroxide, but both the lysosomes and the peroxisomes are recyclers. They're ways of kind of getting rid of debris and kind of keeping our cells and our internal environments kind of clean, quote unquote. So this is our kind of generic animal cell. We are animals, we human beings, and so we do have all of these organelles inside our cells. And part 2 will function and focus on the various organelles in the cell. But let's talk about the plasma membrane first. So this is part 1. Okay. So our plasma membranes are made up of phospholipids. So they have a polar, hydrophilic, water-loving head. It's got a phosphate group attached to it. The phosphate is what makes it kind of okay with the water. And then 2 tails of fatty acids. Well, the fatty acids are hydrophobic, water-fearing. They don't mix well with water. Since both the fluid inside the cell, the intracellular fluid, and the fluid outside the cell, the extracellular fluid, are primarily water-based, because we're primarily a water-based organism, we end up with a plasma membrane that is a bi-layer of phospholipids, with the heads facing out and facing in, facing the water because they're hydrophilic, and then those hydrophobic, water-fearing tails are then on the inside of the plasma membrane. Although the phospholipids are our major component of our plasma membranes, they are not the only component of our plasma membranes. We actually think of our plasma membranes as a fluid mosaic. So the fluid suggests that this is not a rigid, like stiff membrane. It is, stuff can kind of move through it. It is permeable, semi-permeable, selectively permeable, because of that hydrophobic nature of the interior of the membrane. Stuff that is lipid-based can move through our plasma membranes really well, whereas stuff that is non-polar, stuff that is polar, more kind of water-based, doesn't charged, for instance, like an ion, doesn't quite move as well through our plasma membranes. The fluid mosaic also suggests to us, or at least it should, the mosaic part, is that it's not, if you've ever looked at one of those like mosaic pictures, right, it's not just one thing, right? It's lots of little things. And so we do have several different components to our plasma membranes. Yes, of course, the main component is that phospholipid bilayer, but we also have cholesterol, another lipid in our plasma membranes. Our bodies actually make cholesterol as part of our fluid mosaic membranes. Cholesterol helps stabilize the plasma membrane. It is still fluid, it is not rigid, but by kind of packing a couple of carbohydrates, by packing some cholesterol kind of in between the fatty acid tails, the plasma membrane becomes a little less permeable and a little bit more stable and less fluid. We also have lots of proteins embedded in our plasma membranes. They play a huge number of roles for us. Some of them will be what we call a transmembrane protein that will actually span the whole kind of width of the barrier. That's an integral protein. It's internal to the membrane. We also have integral proteins that might kind of sit on just one side or the other. And then we also have peripheral proteins that are literally just on the kind of exterior or interior of the cell, but not kind of within the plasma membrane, kind of just on the edges of the plasma membrane. We also have lots of carbohydrates attached to our plasma membrane. Some of them will be attached to various proteins, so we call them glycoproteins, sugar proteins. Some of them will be attached to some of the phospholipids, so those are glycolipids, sugar lipids. They play a role particularly as kind of an identifier, something that we call the glycocalyx. So these proteins in our cells, in our plasma membranes, just play a huge number of roles for us. They can be transport proteins so that stuff can move in and out of the cell. They might be receptor proteins so that cells can talk to one another. They might be enzymes, kind of speeding up and catalyzing our reactions. They might play roles in cells recognizing other cells, particularly what I call the glycocalyx. They might attach to parts of the kind of cytoskeletal parts of the kind of interior, help move stuff in and out and help maintain the shape of the cell. And they're also playing a role in kind of the junctions between our cells, how cells connect to other cells. So let's look at a couple different roles that our plasma membranes and our plasma proteins play.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood.
So there you saw a transport protein in the plasma membrane. Here we have a receptor protein in the plasma membrane.
Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins.
And then we can also have plasma proteins that are enzymes. And this one happens to be not a plasma protein, but this could just as easily be a protein embedded in the plasma membrane here.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
So just some of the roles of our plasma membrane proteins. Another role that, particularly the plasma membrane proteins that have the carbs attached to them, the sugars attached to them, the glycoproteins, they play a really important role in our body's ability to recognize the cells of our body, and not only for cells to recognize each other, but also for, say, like an immune cell to recognize, oh, this cell belongs to this body. So you can kind of think of the glycocalyx, which is this very specific pattern of glycolipids and glycoproteins. And each different type of cell has a different pattern. So the glycocalyx pattern for a lipid cell, a fat globular cell, would be different than the sugar pattern for, say, a nerve cell. But this very specific pattern, and mine would be different than yours, allows cells to recognize each other, and also allows our body to say, oh, hey, all of these cells belong to me, as opposed to, oh, those cells don't belong to me. That's foreign. That's not self. And so maybe that's something that we need to get rid of, a bacteria, a virus. If you've ever heard of people taking immune suppressants for preventing organ failure and organ rejection when they do organ transplants, that's that glycocalyx pattern coming into play. The glycocalyx pattern of the donated organ has, even though it's say let's the liver, right? My liver has a different glycocalyx pattern than, say, somebody else's liver cells would, and if that person donated their liver cells to me, or part of their liver to me, because I needed it, or say a kidney, then, or sometimes that leads to organ failure because the glycocalyx pattern of the donated organ is different than the recipient's glycocalyx pattern, and so the recipient's immune cells see that kind of a foreign glycocalyx pattern and says, oh, nope, not me, not self, must attack it and get rid of it. Another kind of weird thing that happens with the glycocalyx is cancerous cells. Now, cancerous cells are cells of our body, but they can kind of alter their glycocalyx pattern, and they can do this in such a pace that our body's immune cells don't recognize that, oh, these are damaged body cells. They just don't have the ability to kind of keep up with it. And so the immune cells end up not destroying the mutated cancerous cell, which then unchecks the growth of the cancerous cell. The cancer cells replicate, and you end up with larger and larger tumors. So some of the research that they do with cancer these days is related to allowing your immune cells to recognize kind of cancerous glycocalyx patterns. So now, some of the cells in our body are kind of unattached. They're free, right? Blood cells, red blood cells, white blood cells, they move and roam through our body, and particularly through our blood. Sperm cells are made by the testes, and they have the little flagella tails so that they can swim to fertilize an egg, but they're not attached to anything. But just about all the other cells in our body are attached. They get kind of bound together and tied up together into what we would then call a tissue, which is chapter 4, which is the next chapter. But it's communities of cells, all of the same type, that then kind of come together for a common function. Well, there are 3 ways in which cells come together into tissues: tight junctions, desmosomes, and gap junctions. So the tight junctions and the gap junctions are about permeability in terms of stuff moving from one cell to another cell. Which one do you think is more permeable? If you thought gap junctions, right, gaps, holes, you'd be correct. If you thought the tight junctions made stuff less permeable, right, tight, then that would also be correct. The desmosomes are about kind of anchoring the tissues together, so not so much about moving stuff from one cell to the next, but making sure that the cells kind of stay together. So all 3 cell junction types use proteins, different types of proteins for the different types of junctions. The tight junctions uses these kind of interlocking proteins to really make the cells kind of impermeable to each other, get like a seal around the cell, so that stuff cannot move very easily from one cell to another cell. One of the places that this is really important that we have tight junctions is in the blood cells, in our blood vessels in our brain, right? If something gets into your bloodstream, it can go systemic because you have blood vessels pretty much everywhere. Well, our brain is like super important, and so all of the blood vessels in our brain have tight junctions between their walls, the cells in their walls. It's called the blood-brain barrier, and that prevents stuff that is in your blood from getting out of your blood and into your neural tissue, and is a protective feature for your brain. So tight junctions, impermeable. The gap junctions are the communicating junctions. They're essentially just using these little connexon proteins that makes a little channel that allows stuff to move very easily from one cell to another cell. So these are particularly important in embryonic cells as we go through that kind of developmental process. You'll also find them in smooth muscle in our gut. And you'll find them in the heart, right? So the heart pumps as a unit. Like, we have a 4-chambered heart, and it would be much less efficient if like part of it contracted and then part of it contracted and then part of it contracted. We need the heart to kind of do it all at once. And so the cardiac muscle cells, heart cells, have a lot of gap junctions in them. Cardiac muscle cells and our skin cells also contain desmosomes. So now desmosomes are like welding, spot welds, if you've ever used like solder or Velcro. It uses these little linker proteins, like almost like the teeth on a Velcro. And what the desmosomes do is they really knit together the cells very, very strongly, so that the cells won't separate from each other, even under like high stress environments, like our skin cells exposed to the environment, or our heart, which is constantly beating, 70 beats per minute on average. That's a lot of stress. But despite that, lots of stress, the heart stays together quite well because it has a ton of desmosomes between all of their cells.